Well, welcome back, everybody. This is the second to last chapter for the semester. It's also one of the longest. I'm actually struggling right now to decide whether I want to record this all the way through or do two separate recordings to cover this chapter. There is quite a lot of information involved here. I have broken it up into sections, but even some of those sections are going to be quite long in their own right because of the amount of information that unfortunately we need to cover as part of this chapter. My best advice to you as you are looking at this is to not try and look at it all the way through, irrespective of whether I break it up into two separate recording sessions or do it as a single recording session. You're best advised not to try and watch this all the way through and to take advantage of the breaks. There will be a number of breaks with concept check questions, as always. And I would strongly suggest that you take advantage of those. I may even myself stop the recording for a little while just to step back before I come back and continue recording, even if I decide to go through as a single recording. There is a lot of information to cover here. And unfortunately, the last chapter, chapter 34, is even longer and more energy dense. No, sorry, not energy dense, information dense. So I guess sit back, take your pen and paper, and be prepared to take a lot of notes about a lot of things that you need to be aware of. And by the way, in covering this chapter, we are going to account for about 95% or a little bit more of the entire animal kingdom. That's one of the things that most people don't realize, because our familiarity is with vertebrates. Those are the animals that we spend most of our time associated with, and therefore those are the ones that we, excuse me, we are most familiar with, and are the ones that we generally consider make up most of the animal kingdom if we think about it. But the reality is that that is far from the truth. 95% of the animal kingdom is invertebrate, and a very large proportion of that is not even terrestrial, it's aquatic. So we don't even really encounter the vast majority of the diversity of the animal kingdom because we simply don't live and work in the environments where the vast majority of the animal kingdom actually lives itself. And so we don't really come to terms with the diversity of the animal kingdom until we do a class like this where we are confronted with that diversity. And that means that in order for you to grasp at least some meaningful idea of the diversity of the animal kingdom, we have to spend this time going through just a small part of it. We're not even going to touch on all of the phyla that make up the animal kingdom. We're just going to look at the major ones. We will talk about some of the really small ones just because they are of interest and are of curiosity value. But the clades that we will talk about most are going to be those that are the major clades, the major phyla in the animal kingdom. So that being said, let's start at the beginning. Always a good place to start. So as I said, as I've just indicated, about 95% of the animal kingdom is made up of organisms that lack a backbone, invertebrates. You may recall when we were doing chapter 32 previously that I mentioned there are some 35 to 36 phyla. And of those phyla, all but one are strictly invertebrate. And even the one phyla that is not strictly invertebrate is 2/3 invertebrate, because that happens to be our own phylum, phylum Chordata, which contains 3 subphyla, one of which is subphylum Vertebrata, our own, but the other 2 subphyla, Cephalochordata and Urochordata, are both invertebrates, as you'll discover when we do chapter 34 next time. And so the invertebrate side of the animal kingdom is overwhelmingly the majority of the animal kingdom. And that's why it behooves us to spend at least a little time taking a look at that side of the animal kingdom. Otherwise, we simply ignore the vast majority of the animal kingdom. So this is a phylogenetic tree that tries to outline where we think the current phylogeny of the animal kingdom lies. This is the best hypothesis that we have right now, based largely on DNA sequence analyses. Now, with more genes being sequenced every day and every week, it is likely that we will either reinforce this hypothesis or we'll have to rewrite it. And one of the things that I've actually had to do in prepping this chapter and the next chapter, chapter 34, is to go back and take a really good look at what I've got in the presentations and take a look at what the latest information is in the textbook, and also double check that against my own sources and resources to make sure that I'm giving you the best information that I can based on the phylogeny that the textbook is using and what my resources and my sources are saying about the latest thinking about the animal kingdom. So this phylogeny that comes straight out of the textbook is more or less considered to be the best hypothesis for the phylogeny of the animal kingdom. So we do have a last common ancestor of all animals at the base of the tree. And so you see that located here. Here is the last common ancestor. The most basal group of organisms to the animal kingdom are the sponges, and we talked a little bit about why we still consider those to be basal, even though some people have tried to change the relationships at the base of the tree recently. The consensus is that porifera, the sponges, do rightly belong at the base of the animal tree. They are the most basal group of animals, and so remember that the term for animals is metazoa. So this is the phylogenetic tree for the metazoa. And so when we talk about animals with tissues, we talk about the eumetazoa, true animals or true tissues, which is the vast majority of the animal kingdom. Then we have those that are radial in symmetry, and these have traditionally been referred to as the radiata. I don't see that term being used very much anymore as a clade name, but it is an old name for the radially symmetrical clades, the cnidarians and the ctenophorans. And then we have the bilateria. And just to put this in context, remember the radiata have two tissue layers. They're diploblastic. But just to add in the information that is already here from the textbook, just to make sure that we've got all of that in place. Then amongst the bilateria, we used to split them into the protostomere and the deuterostomere, as I outlined in the previous chapter for chapter 32. But we now do not recognize the protostomere as a valid clade. It is just a description of a series of developmental steps that differentiate those that have the protostomate developmental form from those that are the deuterostomere. And remember, deuterostomere is still considered to be a valid clade. So once we recognize that the protostomere are no longer valid clades, we then have to decide what we're going to do with all of those. Those have been split up into two clades, again on the basis of initially DNA sequence analysis, but also some shared features that are found within the two clades, and we outlined those last time in chapter 32. So the lophotrochozoans, remember, either have a lophophore, a feeding organ, or have the trochozoan larval stage. And even though that creates arguably two separate groups of organisms, we know that they are united by DNA sequence analysis, and they do form a valid single clade. And then we have the ecdysozoans that are, again, grouped as a valid clade on the basis of DNA analysis, but also because they share a common feature, an external cuticle or an external semi or actually rigid layer around the outside. We refer to that as a cuticle, and that needs to be shared periodically as the animal grows, and that process is called ecdysis. And so we have this group called the ecdysozoans that share that common feature. And then we have the deuterostomes, the deuterostomia. There are three major clades. We will touch on two of them as we go through this presentation, the echinoderms and the hemichordata. And even though the textbook does not do a really strong description of the hemichordata, I will touch on them briefly, and we'll talk at length about the echinoderms right at the very end of the chapter. The chordates, well, the chordates, including the vertebrates, are the subject of chapter 34. So basically there's a single paragraph saying we'll talk about the vertebrates and the chordates in the next chapter, right at the very end. So that lays out, based on this phylogenetic tree, where we're going as far as this presentation is concerned. And there's a lot of information to cover, so let's get going. So these photos actually give you some examples of the types of organisms that you might be familiar with from some of the clades. You've almost certainly seen sponges. Certainly in the lab exercise, you had an opportunity to investigate some photos of sponges. The cnidarians and the ctenophorans show radial symmetry, diploblastic, two tissue layers, and we'll talk about the cnidarians at least at length. The acelioworms we're not going to get into very much. They were once considered to be planarians, part of the Platyhelminthes, but they are now separated out into their own phylum because they, on the basis of genetic analysis, DNA analysis, actually form a separate clade that is more basal than any other of the bilaterians, and so they are placed at the base of the bilaterians. The placozoans are an unusual phylum in that there is only a single species known. It hasn't been seen very frequently. It's made up of a bilayer of cells, and they reproduce by binary fission. Apart from that, there's not very much known about them. One thing I do know is that the first example was identified crawling up the inside of the glass case of an aquarium in a state aquarium in Europe. That was where the first individual was identified. It hasn't been seen very frequently. We don't know what the diversity is. We don't know if this is the only species that belongs to this phylum, whether there are other species out there, but certainly this phylum is recognized on that basis. So these are the basal clades. And when we get to Lophotrochozoans, we will talk about Platyhelminthes, the flatworms. Cynodermata is a new phylum created by bringing together two pre-existing phyla, so the phylum Rotifera and the phylum Acanthocephala. These are the acanthocephalans. The acanthocephalans are now recognized to actually be very closely related to at least one of the classes within the old phylum Rotifera, and more closely related to those than they are to anything else. So the general thinking is that the acanthocephalans are highly modified rotifers, and at least at the DNA level, they do form a valid clade. So taxonomists have created this new phylum name, Cynodermata, to cover both the rotifers and the acanthocephalan. We'll talk a little bit about those as well. Then we've got the ectoprocta and the brachiopoda. These are the two lophophorate clades because they possess this organ, the lophophore, for feeding purposes. They are very different types of organisms, but they share that common feature. So these are the lophophore or lophophorate clades within the lophotrochozoans. And then we have a number of other clades that belong to the trochozoan branch or side of this clade, having that common larval form. So we have the gastrotricha. These are another type of worm-like creature. Worm, by the way, is simply used for any group of organisms that has a long, narrow body. It doesn't imply anything more than that. So there are lots and lots of worms, but that's because there are lots and lots of type of organisms that have this long, narrow body plan. So the gastrotricha are another type of worm. We won't talk very much about those. We're not even going to talk about cycliophorans other than this brief introduction to them. There is, again, a single species known. It was identified on antennae associated with a Norwegian lobster. This is the only location where this species is known to exist, and this is the only species known to exist within this phylum. Is there the possibility of other members of the phyla out there? Sure. But where do you go looking? These have a very unusual body plan. They are very, very specific about their location, where they're found. And so it is likely that we're not going to find any other members of phyla unless we get that specific in searching for them, or we accidentally come across them at some point in the future. Certainly finding this species was an accident. There was no deliberate plan to go out and find it because no one knew that it existed. It was discovered by accident. Then we have another clade, Nematea, otherwise known as ribbon worms. These really do match the description: long, thin body plan. Some of these can be up to, I think the longest one on record is about 20 meters in length, which puts it near the top of the list for the longest organism on the planet. These are generally burrowing worms. They live amongst the sediment on the floor of the ocean, burrow in amongst the sediment. We're not going to talk very much about them other than that. Annelida we will talk about. These are the segmented worms, and it's really important that you recognize these as being distinctly different from the vast majority of other worms, because Annelida have this segmented body plan that other worms do not have, and that's very frequently overlooked. And actually, one of my concept check questions a little bit later in the presentation actually addresses the similarities and differences between annelids and nematode worms, which are the two that we most commonly compare. Then we have the mollusks. This is one of the two largest clades that we will address within the animal kingdom. The other large clade is Arthropoda, which is even bigger because of one of the clades within Arthropoda, the insects, or as we now recognize them, crustaceans. We now know that, or believe that what used to be called crustaceans and insects now actually form a single clade. And so there has been a new clade name generated called Pancrustacea, which includes all the previous organisms classified as crustaceans and all the previous organisms classified as insects into a single clade, Pancrustacea. So these are the two largest clades, and historically in the fossil record, and I guess over evolutionary time, these are the two clades that in marine environments, at least, have struggled for dominance as the top predators in the environment. And it's gone backwards and forwards. Fossil record clearly indicates this, that at times mollusks have been the top predators, and at other times arthropods have been the top predators. That all changed with the rise of vertebrates. Vertebrates displaced both of them. And so now, except in very unusual circumstances with very specific ecosystems, it is vertebrates that are typically the top predators in ecosystems. But there are a few ecosystems or habitats that are still dominated by invertebrates as top predators. We may touch on a couple of those along the way as we go. Then we have the ecdysozoans. The loriciferans are a group of parasitic ecdysozoans that attach themselves using this structure to the organisms that they're parasitizing. They can draw themselves into this protective covering. Apart from that, we're not going to talk about them very much. In fact, we're not going to touch on them again. The loricifera are just a small clade. A clade that we will touch on a couple of times are the onychophorans, otherwise known as the velvet worms. The reason why these are important to us is because these are considered to be a closely related clade to the arthropods, and they have been used historically as an outgroup to define arthropods and also to try and understand the origin of arthropods, their phylogenetic origin. And so we will touch on them a little bit. Their body plan is very different from arthropods. It's not segmented. They are basically pretty uniform from anterior to posterior, unlike arthropods. And that actually gives us some room to think about the origin of arthropods because of the highly segmented and specialized nature of the arthropod body plan. More of that to come. Then we have the priapulan worms. If it looks a little bit like a penis, then congratulations, because the common name for these worms is penis worm. They are named for the Greek god Priapulus, the god of fertility, and he was recognized because he had a very large penis. It dominated his body plan. And so I guess when people saw these and the structure of these worms, the name came to mind immediately. These are burrowing worms. They burrow in the sediment. And again, we're not going to touch on those very much at all. This is basically all we're going to talk about. Then we have the nematode worms, otherwise known as roundworms. This is a fairly large clade, and it's a very important clade because nematode worms are parasites of vertebrates, including humans. They are also present in the soil in very, very large numbers, and Florida is a particularly good habitat for nematodes. They are a major agricultural pest, and it's one of the reasons why Florida agriculture relies very heavily on greenhouses these days. And we may touch a little bit on that when we talk about the nematodes. Then we have the tardigrades, otherwise known as water bears. These are cute little animals. People refer to them as water bears because they look very bear-like in form and they waddle along much like a bear. And these guys are more or less indestructible. We don't really know what it takes to kill these. You can drop their temperature down to -200 degrees Celsius or even further. They go into a stasis. They produce specialized proteins that protect their internal body structures from freezing damage. When you bring them back up to room temperature again, they revive and go about their business as if nothing had happened. They've survived the vacuum of space. They've survived large doses of radiation. They've survived severe dehydration. As I said, we really don't know what it takes to kill these. And so as a consequence, they are a major source of research interest because people want to know what it is that allows them to protect themselves under all of these harsh conditions. And when they return to normal conditions, to shrug it off as if nothing had happened. And we know that a part of it is because they do produce these protective proteins, the ones that I mentioned when I talked about the low temperature survival. we do know that they produce these specialized proteins that serve to protect them from the damage that otherwise we would expect to see as a consequence of these very harsh conditions that they are only too easily able to survive. So they're subject to a lot of research as a consequence. Then we have the single biggest clade. You'll notice that it's given as 1,000 species, sorry, 1 million species. That's 1 million species known to science. In other words, someone's taken the time to, let me erase that. All this means is that someone's taken the time to collect specimens, analyze it, write it up, and publish a paper describing the species. So there are about 1 million species of insects that are currently known to science that have been described in the literature. The actual true number is anybody's guess. I've seen numbers as high as approaching 10 million. I think the number that most people are happy with as a consensus number is somewhere around 3.5 to perhaps 5 million species of arthropods, most of them being insects. And what's remarkable about that number is that as far as species of all animals that have been described to science, it's around 3.5 million. So to have 1 million species described by scientists out of one group, not just the arthropods, but the insects, and to think that there may be at least 3.5 million species of insects out there. And we've only ever described about 3.5 million species total, is quite remarkable. But it just tells you just how successful arthropods, and particularly insects as terrestrial dwellers, have been. And one of the things that we will discuss when we talk about the arthropods is just why they are so successful. There has to be reasons why, and we will get into those reasons. Then we have the last group, the Deuterostomia. And here are the 3 major phyla, the Hemichordata, which we're not going to talk about very much. These are sometimes known as acorn worms. The name Hemichordata refers to the fact that they do share 2 features that chordates have. They have a notochord and they have pharyngeal slits. So they have 2 of the 4 shared features that all chordates have, hence the name Hemichordata, because they have some of the features. Then you have the Echinodermata, a very diverse group of organisms, 5 phyla with 5 classes within the phylum, or clades within the phylum, because people are going backwards and forwards as to where the clade should fit in the biological hierarchy. Some of these are very, very ancient and have remained very much unchanged over evolutionary time. And basically, when we look in the fossil record, we see organisms that are easily identifiable because they are essentially identical to living examples. So we'll talk a bit more about those when we get to the echinoderms. And then we have the chordates. The example shown here is a tunicate. It is belongs to Urochordata, one of the 2 invertebrate subphyla within the phylum Chordata. Interestingly enough, this is the one that is, Urochordata is the clade that is considered to be most closely related to our own subphylum, Vertebrata, even though the tunicates don't look like they have bilateral symmetry as adults. In their larval stage, they most certainly do. They have all the characteristic features of a bilateral organism, and they share the 4 common traits, chordate traits, that we associate with chordates. And so they are bilateral invertebrate chordates. Why are they the most closely related to vertebrates? Based on DNA analysis, that's where we put them. And we can, by looking at both Urochordata and Cephalochordata, the other invertebrate subphylum of phylum Chordata, see where the origin of vertebrates actually lies. And we will talk a little bit more about that and the role of Hox genes when we do chapter 34. So this is the diversity of major phyla, and I've identified the ones that we are going to talk about as we make our way through both this chapter and the next chapter. Remember, this chapter is about everything except the chordates. So all of the phyla that I've identified in the previous slides as ones that we'll talk about, that's what we have to discuss in this presentation. So as you can see, there's a lot to discuss. So let's start with the most basal group of the animal kingdom. These are the sponges. They used to be split into two phyla called the Calcarea and the Saliceae on the basis of the structures known as spicules, these rigid reinforcing structures that are inside the body of the sponge. If the spicules are made out of calcium carbonate, then the sponges were assigned to Calcarea. If the spicules were made out of hydrated silica material, then the sponges were assigned to Saliceae. And so that's how they were split out. I don't see much evidence of those two phyla being currently recognized. Certainly the textbook at the moment does not address these two phyla as being part of the larger group Porifera. So my guess is that there is still some question as to whether we need to recognize two phyla of sponges or whether they're going to be merged into a single phylum yet to be named. Irrespective, the vast majority of sponges are marine, but there are a number, about a dozen or so sponges that are freshwater sponges. Generally, they are quite small compared to their marine relatives. What's separates sponges from the rest of the animal kingdom is that they lack true tissues, and in the absence of true tissues, obviously no organs and no organ system. So that note should be self-evident. All sponges are filter feeders, and they are capable of filtering very large volumes of water per minute. I've posted in the resource page in Canvas a video from an old series that's about 20 years old now called The Shape of Life. Even though some of the information in the videos from The Shape of Life that I posted as part of both this chapter and chapter 34 is getting a little bit dated now, a lot of it is still quite valid, and certainly in terms of being able to see the animals in their native habitats, following through on their native behaviors, is somewhat useful. Actually is very useful. So I would recommend that you take a look at some of those videos, and the one on sponges shows you just how much water they are perfectly capable of filtering. And the reason why this is important is because the action of sponges in filtering the water maintains water quality. This is particularly true in ecosystems like coral reefs, where the sponges are a very important part of the ecosystem because their ability to filter the water and keep all the sedimentary material out of the water maintains good water quality for the corals. Because as you'll see when we talk about the cnidarians and we focus on coral polyps particularly, coral polyps, a lot of coral polyps are dependent on symbiotic photosynthetic organisms, mostly dinoflagellates. And for those organisms to do photosynthesis, they need clear water. And so the sponge's ability to filter the water and keep the water quality high and keep the water clear is important for the success of the coral polyps and the success of the coral reef. And remember, coral reefs are the areas or the habitats, ecosystems in marine environments that represent the greatest level of biodiversity. You could think of them as the rainforests of the ocean. So they're really, really important. If we take a look at the typical body plan of a sponge, you find that there is an outer layer, the epidermis, which is made from ectoderm type tissue. Then you have a sponge-like layer called the mesohyal, which has the spicules embedded in it, and also a group of cells called amoebocytes. These are mobile cells, and they have a huge importance to the success of the sponge. And then on the inside of the sponge is a layer of cells that represent the feeding cells called choanocytes, which, if you think they look very much like choanoflagellates, that's good, because they are, to all intents and purposes, cells of that nature, but they're part of a larger unified organism, or somewhat unified organism, as the case may be. So the choanocytes capture food, just as the choanoflagellates capture food, through a water current being filtered through the net-like arrangement between the tentacles of the collar. The food particles are phagocytosed and then either utilized by the choanocytes or passed on to the amoebocytes. Now, the amoebocytes are mobile inside the mesohyal, so they can transfer these food particles, or food vacuoles, to the other cells that make up the sponge. So you've got the cells of the epidermis, and you've got the porocytes, the cells that form the pores that allow the water to flow from outside the sponge into the inner part of the sponge, the spongiocele. What the amoebocytes also do is maintain totipotency. Now, you may recall from a previous chapter, that would be chapter 47, that cells that are totipotent have the ability to become any of the cell types found in the body of the organism. And so amoebocytes are really important to allowing the sponge to reshape itself in response to environmental changes. So if water currents change, if there are changes, if the sponge is dislocated from its anchor point and needs to settle down again, it may need to reorientate the sponge body. It is the amoebocytes that provide the source of new cells to be able to do that. So amoebocytes and the daughter cells of amoebocytes can become any of the several different types of cells that are found in the sponge. Amoebocytes also can undergo meiosis to give rise to the gametes. And the amoebocytes produce both the eggs and the sperm within the mesohyal. The eggs are retained within the mesohyal, and the sperm are released into the spongiocele and exit the body of the sponge through the opening called the osculum. And the sperm then are released into the water column. They will be drawn into the body of a sponge of the same species eventually, at least that's statistically what is expected to happen. And when they are in the body of a sponge of the same species, the sperm egg recognition proteins allow the sperm to fertilize the egg cell within the mesohyal, and that gives rise to a zygote, which develops into a larval stage, which enters the water column and may drift in the water column for a short period of time until it settles down on the substratum and starts to develop into a new sponge. What is interesting is that the relationship between the cells that make up a sponge is somewhat loose, and yet they can still recognize each other. If you take, for instance, a small freshwater sponge, and again, I can't remember whether I posted a video into the resource section or not, but there are several videos that demonstrate this, which you should be able to find on YouTube. If you pass a small freshwater sponge through a very fine mesh and break it up, so that basically you've got small clumps of cells rather than the intact sponge, one of two things will happen depending on the nature of the sponge. If these cells are able to recognize each other, they will come back together and reform the sponge body again. If, however, the connections between the clusters of cells are a little more loose and they're unable to recognize each other, then those clusters of cells will develop into new sponges, relying on the amoeba sites as totipotent parental cells to give rise to all the necessary replacement cells to build out the sponge body. And so again, the role of the amoeba sites is multiple. They are transporting cells because they are amoeboid, they can move around in the mesohyle. They are totipotent, so they're functioning as the equivalent of stem cells for the sponge. And they also can undergo meiosis to give rise to the gametes, both eggs and sperm. So they are multifunctional cells within the body of the sponge. So, time for our first break, and I am going to take these breaks pretty seriously and actually stop the recording at each one of them, because as I said, even though some of the sections are relatively short, some of them are going to be long because of the information that's in there, and this whole presentation is going to be long. So breaks along the way are going to be quite important. So I am going to break the recording and then come back on the other side and talk about this concept check question. So the question is, explain how sponges feed. Why is this important for ecosystems like coral reefs? I will see you on the other side of the break with the answer.